I'm Javairia Haq, and I'll be talking about point mutations. A point mutation is a genetic mutation where one nucleotide base is substituted for another. It's important to remember that mutations are completely random. There are two types of point mutations. The first one is called transition, where the base is changed from a purine to a purine or from a pyrimidine to a pyrimidine. The other type is called a transversion, where a purine is changed to a pyrimidine or a pyrimidine to a purine. Here are the possible base changes. Point mutations have four different effects at the protein level. The first is missense mutations, which changes the amino acid into a different amino acid. We are given the following DNA coding strand. The coding strand has the same base sequence as the mRNA strand, except each thymine now becomes a uracil. We can then use the codon dictionary to see which codon codes for which amino acid, and we'll stop translating once we reach a stop codon. In this example, a point mutation has occurred that caused the guanine to be changed to a cytosine. This is a transversion since the purine changed to a pyrimidine. As a result, our mRNA strand has changed and a different amino acid has been created, going from alanine to proline. This is an example of missense mutation. The second type of effect is called silent, which means the change in the nucleotide base did not change the amino acid. As shown, we still have our original DNA strand. However, a point mutation causes a thymine to change to a cytosine. This is a transition since both are pyrimidines. The mRNA trinucleotide has mutated, but both GCU and GCC, the mutated trinucleotide, code for the amino acid alanine. The third type of effect is called nonsense, which changes the codon so that it becomes a stop codon instead. In this example, the cytosine in our original DNA strand has mutated to a adenine, so this is a transversion. The mutation causes the mRNA codon to become UGA, which is now a stop codon, and it stops translation. The fourth effect is called read-through, which changes the stop codon to a codon that codes for an amino acid. In the last example, the point mutation changes the thymine in the original DNA coding strand to a cytosine. Both are pyrimidine, so this is a transversion. The mutation causes the resulting mutated mRNA trinucleotide to become CGA, and this codon codes for the amino acid arginine instead of being a stop codon. This causes translation to keep going, and we end up with a longer protein.